Now that you are much familiar with the immune response to bacteria, don't you think we shall discuss that what exactly is responsible for generating this response? Or how can our immune system recognize these bacteria as foreign and initiate an immune response? Though you all have knowledge about the basic structure of bacteria, but let's have a look at it once again, where we would specifically focus on the role of each structure in antigenicity. So an important structure of a bacterial cell is a cell wall, present outside the plasma membrane. Bacterial cell walls are rigid and protect the organism from differences in osmotic tension between the cell and the environment. Do you know that the cell walls of gram-positive and gram-negative bacteria are a bit different? Well, gram-positive cell walls have a thick mucopeptide called peptidoglycan layer present outside the cell membrane. In addition, gram-negative bacteria have a second membrane with lipopolysaccharides, also called LPS or endotoxin, inserted in it. Now, this peptidoglycan, or PG for short, consists of a chain of alternating N-acetylglucosamine, or GLC-NAC, and N-acetylmuramic acid, or MUR-NAC, units that are linked by a beta-1 4-glycosidic linkage present between these two glycans. Well, it is in the peptidoglycan that the enzyme lysozyme acts by attacking the N-acetylmuramic acid and acetylglucosamine links. Now, lysozyme, you can better guess through the name that these are the enzymes present in lysosomes. As I already mentioned, that they attack N-acetylmuramic acid and acetylglucosamine links, so you will also come across another name for these lysozymes, that is muramidase or N-acetylmuramide glycan hydrolase, because they are hydrolyzing this particular linkage. Well, bacterial cell walls are powerful inducers of inflammation, largely through their ability to activate the toll-like receptors of innate immunity. Now, toll-like receptors, or TLR, are a class of pattern recognition receptors, or PRR, that initiate the innate immune response by sensing pathogen-associated molecular patterns, or PAMP, for early immune recognition of a pathogen. And toll-like receptor 2 has been shown to recognize peptidoglycan along with several other PAMP. Now, another important structure of bacteria are flagella, that allow organisms to find sources of nutrition and penetrate host mucus. These are the main agent of bacterial motility and contain highly antigenic proteins. The H antigens of typhoid being a good example, which give rise to immobilizing antibody. H antigen is also known as flagellar antigen. Some flagellar proteins activate the toll-like receptor, TLR5, as these TLR5 recognizes an evolutionarily conserved site on bacterial flagellin that is required for flagellar filament assembly and motility. So the bacterial flagellum affects bacterial virulence in various ways. That is by providing motility towards host targets and by promoting adherence and invasion, promoting early biofilm formation and thus bacterial survival, secreting virulence factors, and triggering the adaptive and innate immune defense. Next up are pili, also known as fimbridae. Well, the pilus is a hair-like structure associated with bacterial adhesion to the host cell and related to bacterial colonization and infection. Pili are primarily composed of oligomeric pilin proteins, which arrange helically to form a cylinder. Now, the antibody can prevent the adhesion through pili to host cell. One example being the IgA antibody against gonococcus. The fimbrial antigens are often immunogenic, but vary between strains, so that repeated infections may occur, like in Neisseria gonorrhoeae. Also, Escherichia coli that causes urinary tract infections bind to mannose receptors on ureteric epithelial cells by their P. fimbriae. Now, the outermost structure of bacteria, the capsule. Many bacteria owe their virulence to capsule, which is in fact a polysaccharide layer that protects the cell from phagocytosis and desiccation, where most of them are large, branched polysaccharide molecules, but some are protein in nature. Many of these capsular polysaccharides, and also some proteins from flagella, are T-independent antigens. Now, what are these T-independent antigens? Well, antigens that can stimulate B cells without the help of T cells are thymus-independent or T-independent antigens. Some examples of capsulated bacteria include pneumococcus, meningococcus, and Haemophilus specimens. Next, we have a group of soluble proteins that are secreted by the bacterium, enter host cells, and catalyze the covalent modification of a host cell component to alter the host cell physiology. Yes, I am talking about exotoxins. Exotoxins, in contrast to endotoxin, are diffusible proteins secreted into the external medium by the pathogen. While most pathogens secrete various protein molecules that facilitate adhesion to or invasion of the host, many others cause damage to host cells. Now, the damage may be physiological. For example, cholera toxin promotes electrolyte and fluid excretion from enterocytes without killing the cells. Or pathological, where the toxin, like diphtheria toxin, inhibits protein synthesis and induces cell death. In addition, there are proteins collectively known as aggressins that help the bacteria to spread by dissolving host tissue. Now, after discussing various components of bacteria, I think a significant condition worth mentioning here is sepsis. Occasionally, uncontrolled systemic responses to bacterial infection develop, which can lead to rapid life-threatening disease, a toxic shock. Such responses are still an important cause of death after major surgery. Well, sepsis happens when an infection you already have triggers a chain reaction throughout your body. It is believed to be associated with an exacerbated release of mainly pro-inflammatory cytokines, such as tumor necrosis factor, or TNF-alpha, interleukin or IL-1, IL-6 or IL-12, interferon gamma or IFN gamma, and macrophage migration inhibitory factor or MIF. This brings us to the end of this section. Now, it is fortunate that most of the bacterial structures are strongly antigenic and antibody overcomes many of their effects. And this forms the basis of the majority of antibacterial vaccines.